Arsenic. Arsenic is a toxic carcinogenic metalloid that occurs naturally in the terrestrial and the aquatic environments. Arsenic exposure is mainly through the contaminated groundwater used for the drinking purpose and has widely been associated with the detrimental health effects. Arsenic exposure affects more than 200 million people worldwide, and it has emerged as a major public health concern in India and Bangladesh over the last few decades. Now, it is a well-established fact that not just drinking water but irrigation water contaminated with arsenic is an important source of human arsenic exposure through the water-soil crop transfer. Continuous buildup of arsenic in the paddy soil is mainly from the use of contaminated irrigation water. And rice production systems are of particular concern because the flooded soil conditions are the most favorable for arsenic uptake in rice. In Asia, rice is the basic staple food for majority of the population, including the region's 560 million people. On the other hand, the Asian region also accounts for 90% of the global rice production and consumption. So, minimizing arsenic intake from the food consumption is a key aspect of the public health response in the arsenic contaminated regions. While drinking water arsenic has a permissible or a guideline value, quantification of the guideline values of the soil arsenic is limited. We have previously used a meta-analysis approach to predict the maximum allowable total arsenic in the soil based on the decision tree and the logistic regression models. The models were defined using the maximum tolerable concentration of arsenic in rice grain as per the codex recommendations. The logistic regression predicted 11.75 and the decision tree 14 mg per kg of total arsenic in soil as the allowable limits. In our present study entitled Determination of the Bioavailable Arsenic Threshold and Validation of the Modeled Permissible Total Arsenic in the Paddy Soil using Machine Learning, we validated the decision tree and the logistic regression models for the total arsenic using the test datasets derived from purposely collected field samples from the arsenic contaminated sites. And we also estimated the threshold limit for the bioavailable arsenic in the soil above which the grain arsenic may surpass the codex recommended values. The decision tree model performed better than the logistic regression model in terms of the model parameters, and therefore the total arsenic in the paddy soil of 14 mg per kg could confidently be used as an appropriate guideline value. This box plot shows a comparison between the three test datasets used for the purpose in terms of the category of grain arsenic that is less than MTC and more than MTC with respect to the soil total arsenic and the limits predicted by the logistic regression and the decision tree model. The blue points below the red line representing the 14 mg per kg of total arsenic in the soil from the decision tree model represents the instances at which the rice grain arsenic was more than MTC. These particular instances are due to the fact that in addition to the total arsenic in the soil, the bioavailable or the bioaccessible fractions may be playing a significant role, leading to a high uptake of arsenic in rice grain. And this warrants further investigation considering the other soil parameters like the pH, organic carbon, the available iron and the phosphorus, and which leads to the next part of our research paper. The field data were used to predict the concentration of the bioavailable arsenic in the paddy soil with the help of random forest, the gradient boost machine and the logistic regression algorithms. Here, the category of the grain arsenic that is less than MTC and the more than MTC was considered as the dependent variable, and the bioavailable arsenic, the total arsenic, the pH, organic carbon, available phosphorus, and available iron as the predictor variables. From the better performing logistic regression model, it was observed that the bioavailable arsenic, the total arsenic, the available iron, and organic carbon were the significant variables for the grain arsenic concentrations. From the partial dependence plots and the individual conditional expectation curves, that is the ICE plots of the better performing LR model, it was observed that 5.70 mg per kg to be the limit for bioavailable arsenic in soil. Lastly, in spite of the uncertainties and inherent limitations of the models brought on by the lack of sufficient and quality field data, this is a novel way of predicting the grain arsenic content. As the models have been developed using a specific set of data from specific geographical regions, it would be naive to think that they could be applied to all the contaminated rice growing sites globally. However, testing and fine-tuning the models with more field data will enhance their applicability and will serve as a protocol to derive site-specific regulatory limits.